So, welcome back to everybody. Thank you to be here again for this long day. So it's the official welcome for the third React Congress 2016, the International Congress on Research of Rare and Orphan Diseases. We started in 2012. So the idea of the opening ceremony, it's to give you a more general overview. When I say you, I'm thinking about research and scientists. So for that reason, I will really briefly introduce what maybe for you it's obvious, but maybe that you have to remind at least one per week when you are in the lab, on the bench. So Blackstone Foundation amplify collective action against rare disease. Fortunately, we are not the only one. We are more focused on research, as you probably understood, and we are trying to work early in collaboration all around the world with all the institutions that exist. But briefly, I want to introduce you again what is a rare disease, because we are talking about science, but I don't know if all of you are aware about the definition of a rare disease. In Europe, it's 1 out of 2,000, 1 person out of 2,000, but this is just a numerical definition. I think it's not enough. We have to remember that those diseases are chronic, debilitating, are progressive, degenerative, and unfortunately very often are life-threatening. Those are just an infographic with many numbers, but if you see this picture, of them in comparison with rare disease people. And if you put that on the numbers of international population, all rare disease patients combined together is the third population in the world after China and India. For that reason, rare, it's unfortunately not the best term that we have to use. And if we are looking closer, 80% of the population is suffering from rare diseases. We estimate that 75% of them are children, because of course most of them are from genetic origin, and 30% will not survive the first year of life, and the other 35% not 5 years old. So those numbers are really impressive, and at least personally, I really want to thank you researchers that are working on those diseases, because those people deserve a treatment. So for the OMS definition, there are 13,000 diseases that are mentioned in the OMS numbers, which 8,000 are rare diseases. 80% of them have a genetic origin, as I said before. For that reason, most of them are occurring in children. And this was something that was published by Genes. If we put together AIDS and cancer, rare disease impact more people than those diseases. And what we do? First of all, the React initiative is composed by a human platform where people can present state-of-the-art, discuss the results, and most of them exchange ideas and start new collaboration. So start to collaborate with other people. And the React Congress was the first answer, and I'm really pleased this year to say that this was possible also with a partnership with Daria that was very, very nice to collaborate with you, and you participated a lot in creating this third edition. The second activity that we developed is the React community, which in fact is the same idea of the Congress. Instead of bringing you here, we are trying to let you meet online. And the idea of the community is really to again, to learn. I will not say it's you. To meet, to share, and most of it to support, because we also offer the possibility to start crowdfunding project in rare disease research. And we are going to have, I'm sure, a fabulous presentation of Heather on Saturday on that topics. And we want to start an international digital awareness campaign. As Luigi this morning used it very well, he's a revolutionary researcher, but we are all revolutionary people, and we can all do something something strong for scientific research. For that reason, on the social part, please tweet about that, share the knowledge, share the presentation and the scientific information that we will have during those 4 days. And also we decided today to launch a petition which aims to increase the support, the general support, international support on research on rare and orphan diseases. So please go on change.org, sign it, share it, and again, then in 1 year time we are going to deliver it and try to really get more support for rare disease research. So again, Black Swan Foundation aims to empower a community that deserves to be heard. In fact, the aerobics and rare disease sentence of this year. And you can find all the information that I presented today on our website. So I thank you for your attention. But now, the most important part, and I'm pleased to introduce you Professor Christopher Austin from NIH. He's in charge of NCATS. It's the director of NCATS, and he will present Catalyzing Translational Innovation. So, Professor Austin, the floor is yours. Thank you.
Thanks. It's great to be here. And I'm going to run through very quickly with you some of the things that are going on at NCATS. And for those of you who don't know what NCATS is, that's okay. We're relatively new still. We're younger than React. We're only about 4 years old. So let me dive right in here. Do I need to use this? Yeah.
Maybe the other one?
The other one. I need to use Okay. All right. So just okay. Yeah. Okay. This is true with rare diseases, but it's true in all diseases, really. That we, I think, all are excited and troubled at the same time by this paradox that we live in. That we know more about ourselves in health and disease than we ever have, perhaps exemplified by the Genome Project. But we're all painfully aware that what is on the right side, which is an improvement in health, has not in any way kept pace with the really ethical changes that have happened in science in the last 30 years. And this just sort of exemplifies it. You can see this title in the back, but it's a report from the Institute of Medicine in the United States that says it's titled "Shorter Lives, Poorer Health," and it kind of says it all about the changes that need to happen in the way that we do research and deliver care for the promise of what's on the left side to be delivered on the right side. And the kind of bottom line here is that people are unhealthier than they need to be. And funders, and that includes patients, of course, and taxpayers, and funders, both public and private, have become impatient with us. And I think we've become impatient with us, too. So what do we do about this problem? Well, the news is not all bad. That is, things have really changed over the last 30 years. This year is my 30th reunion from graduating from medical school, and things have really changed in that last 30 years. This is the graph of the last 30 years, and this is the number of conditions in OMIM which have a defined molecular basis. So 30 years ago, there were less than a couple of dozen. Now there are well over 5,500 diseases that we understand the molecular basis of. But of course, what we're really interested in is not just understanding them, but treating them. And these are both rare diseases and common diseases. Only about 500 of them have treatment. You're well aware of this problem. So as I often put this, this has been traditionally, and still is, the coin of the realm in academic research. This is actually the report of the original genetic disease that was reported by Linus Pauling in 1949. This is the molecular basis of sickle cell disease. This does not equal this. And often I think researchers and policymakers believe this is true. And we don't need to look farther than to ask, is there a drug directed against the molecular pathogenesis of Linus Pauling reported in 1949, 67 years later now? The answer is no. So it's clear that we need to have a dedicated effort to go from the left side to the right side, and how to do this much more efficiently. I often think about a mom of a child with a rare disease that we work on, who said to me a couple of years ago, you know, I love publications, I love scientific discoveries, but when my daughter is sick, I can't give her a publication. And I think of that frequently, because I think that boils down the problem that's in front of us. Okay, so you also probably know that as an ecosystem, our ability to create new therapies out of all of this incredible motherload of information has actually been getting worse, not better. So over the last several decades. So you all know about Moore's law. I hope if I can find my iPhone, which is now almost too small to find. Here it is. It's in my pocket. Now, when I was in college, again 30 years ago, the computing power of this thing would have taken about half this room. Given the fact that the number of microprocessors you can put on a transistor has increased logarithmically over the last 5 decades. That's why one has seen this dramatic revolution in computer power. But during that time in therapeutic development, we have something called Eroom's law. This is Moore's law spelled backwards. If you haven't read this paper, it's not like reading. It's not good if you're looking for a pick-me-up, because the data are rather sobering. But what you're looking at here is the monotonic decline in productivity of creating drugs over the last 60 years now, starting in 1950. That is, that per $1 billion spent, the number of drugs has gone down produced has gone down by about half every 9 years since 1950. And this is adjusted for inflation. And if you could just look at this from a business standpoint, any business which has dramatically and consistently negative productivity growth is going to go out of business, merge, and or have the products which are produced be exorbitantly expensive. And that's exactly what we see in the biopharmaceutical industry. And one of the interesting things about this is that this graph has continued to go down despite the invention of computers, recombinant DNA, the genome, et cetera. And for those of you wondering where this begins to asymptote towards zero, the answer is about 2050. So unless something changes, by 2050, there will be no more drugs. Or in other words, the cost of a new drug will be infinity because it's a log scale, which is pretty much the same thing. So if there's anything, these two numbers, this distinction between the number of diseases we understand, the number of diseases that need to be treated, you just heard those numbers again, you don't know them, but it bears reciting, because I think the population at large doesn't understand this large number of diseases that we have to treat them, and the fact that our ability to treat them, the ability to create treatments is getting worse, not better. If anything, calls for a dramatic change, not a little change around the edges, not a little incremental change, but dramatic disruptive change. I don't know what does. Okay, so that's one of the reasons that NCAPS was formed. So this NCAPS was formed 4 years ago, and this is its mission to catalyze new ways of doing things. it's fundamentally new ways of doing things that will dramatically increase the speed with which treatments are developed, diagnostics and treatments are developed and deployed. So we're all about new methods, new technologies, new ways of doing things. So I frequently had to define what translation is. At the NIH, you probably know that there are 27 institutes, and those institutes are, for the most part, defined by organ systems or diseases, just like doctors are. So you know we have heart doctors and eye doctors and cancer doctors, et cetera. Well, at the NIH, we have a heart institute, and we have an eye institute, and we have a nose institute, and we have a cancer institute. And it's fairly well understood what those institutes do, because everyone knows what cancer is and what an eye is and what a heart is. But when you ask people, well, you work for the National Center for Advancing Translational Sciences, and this is fitting for this discussion, at the beginning, I was asked, so you have a center which works on linguistics, the science of translation, right? Isn't that what you do? And because that's what translation means to normal people. It doesn't mean this. So we had to define this term, and this is one of the problems culturally that we as a community need to understand, that the average person, at least in the United States, and I think this is true in Europe too, does not even understand that this process exists. If you don't understand the process exists, you're not going to solve it. And so defining what the problem is, why it is so difficult to go from the discovery to the treatment, and this whole process of translation is something that we need to take on as a community. So this is our broad definition. It's the whole process by which it goes from an observation that's made in a laboratory, say a beam discovery, a clinic, sort of, let's say a doc makes an observation in a clinic that he thinks is going to be an insight which could lead to a novel intervention, or a community health, public health workers in the community, to think about the Ebola epidemic or now Zika. Those observations are turned into interventions, usually physical things, drugs, devices, diagnostics, behavioral interventions, that are shown to improve the health of individuals, and then in larger and larger numbers of people, and then eventually in people living in the community, not simply people in clinical trials, because of course we know that the results of clinical trials may not translate to, as a friend of mine likes to say, free-range humans, people who are living in outside the food system. But you'll notice that NIH or NCATS is not the National Center for Translation. Most institutes at NIH, I suspect most of your organizations, do some aspect of this, perhaps many aspects of it. We are the National Center for Advancing Translational Science. So what is translational science? It's the field of investigation that seeks to understand what the general principles are by which this process happens. Our thesis is that the reason for this implacable decrease in effectiveness of developing novel interventions is that we don't understand what the general principles are. And if you have an empirical process, which this is, which translation is for the most part, so it's a almost relentlessly empirical process, trial and error process, that if you have that kind of trial and error process, the likelihood of your getting from beginning to end is exceedingly small. And in fact, that's exactly what we see. For you geneticists in the crowd, I often ask people to imagine, if one is trying to do molecular biology, just imagine trying to do genetics without understanding that A goes with T and G goes with C. Genetics would be empirical. If you're going to make a PCR primer, you have to make 500,000 PCR primers and test them all individually. You don't have to do that because you understand the physical chemistry of that reaction. You don't understand the general principles of translation, and we need to understand them. Interestingly, very few people study this problem as a systematic problem. NCATS and some aspects of the EU do this. Relatively good. So, but it's not just a scientific problem; it's an organizational problem. It's an incentive problem. It's a legal problem. It's an ethical problem. All of these things which can get in the way. Okay, so what does NCATS do? I'm not going to go through all of these, and you'll have these slides, but not surprisingly, we have divided what we do into clinical people, in preclinical, that is before we get to people, and then broad programs to re-engineer the whole process of clinical entry. I'm just going to run through a few of these for you to give you an indication of how we're approaching this problem. So on the clinical side, the largest part of this program is something called the CTSA program. It's a national consortium of most of the well-known academic institutions, medical institutions in the United States, and they work together to improve the way the clinical aspects of translational research are done in the United States, and as well as doing innovative training. And this is the map. If you're familiar with the geography of the United States, you'll know that there are large concentrations of biomedical researchers in New York City, in Boston, around DC, San Francisco, LA, those kinds of things. But in addition to that, you'll notice that there are sites across the country, north, south, east, west, urban, rural, etc. And when I took over NCATS, this was a rather disparate program. That is, that these organizations were not working together. And I looked at this and I thought, wow, what an opportunity. If we could tie all of these centers together with a common informatics infrastructure, a common IRB infrastructure, common contracting, common clinical qualification, we could turn this into a national network for translational medicine. There are about 130 million people taken care of across all of these places. Virtually every key opinion leader in every area of biology and disease is at or affiliated with one of these places. And we are well on our way to doing this now, about 3 years later. And you'll be hearing more about this over the next 6 months to a year as we roll out this capacity. And let me just give you a sense of some of the things we've been doing. In the US at least, doing a multi-site clinical trial, and as you all know, for rare diseases, essentially all multi-site clinical studies. The current system is tremendously inefficient and costly. And a lot of that is due to human subject protection, duplicative human subject protection reviews among sites, things as dull and unscientific as subcontracting and investigator site qualification are things that mean that for a study in the United States, it's routinely 2 years of simply going through paperwork before a study can get started. And particularly in rare diseases, many of those patients will become so sick that they are no longer eligible for the trial or will die waiting for us to do this paperwork. And that's simply unacceptable. It's a solvable problem. And so I'm glad to tell you that, again, within about 6 months, I'm very confident that we will solve this problem in the US. Going from a review time within these institutions of about 2 years, which is what it takes now, down to 2 months. That's the goal. And we're well on our way to doing that. Another problem in the US is that a large number of trials fail to recruit. The numbers are about half of studies which are funded by the NIH fail to get enough patients to recruit. And they often spend 3, 4, 5 years working to recruit actually, so it's half of their people into a trial, expose those people to an intervention with what turns out to be no chance of benefit because they never reach a statistically significant number of people to actually find out if the intervention work. Now, in the era of Facebook and Twitter and unstoppable and unstopping communication, this is not a technical problem. A good friend of ours, many of you probably know, Ron Bartek, who's the director of the Free-Race Ataxia Association, has my favorite story about this, that by tying together, from an informatics perspective, the Free-Race groups, they were able to recruit for one of their most recent clinical trials in 2 hours and 47 minutes. So I have now told our people, I want our trials recruited in 2 hours and 47 minutes. If Ron can do it, we ought to be able to do it too. And again, this is something that this group runs into over and over and over again. This is not a technical problem. It's a cultural problem. It's a sharing problem. There are all kinds of excuses that people throw up that things are incompatible. Don't believe them, frankly. I have not yet run into anything which is not not solvable from a technical perspective. It just requires leadership and will, and these things can be done. So we're well on the way here with creating a nationwide EHR system that allows us to qualify patients and then qualify them for clinical trials. And we've started doing pilot studies to look at this, and it's actually performing exactly as we would imagine. You can imagine if you have 130 million people to choose from or find patients. Another thing which we've been focusing on is that, again, at least in the United States, the way we have trained people for the last 100 years, both scientifically and culturally, is not adapted well to the problems of today. And so there are areas that are non-traditional, such as regulatory science, entrepreneurship, that we're insisting on everyone having training in. We think about training translational researchers the same way as we think about training interpreters or translational people in linguistics. So if there are people, let's say if you're in the United States and you're trying to learn a foreign language, in my case it is French, and then I not only need to understand that language, but I need to understand the cultures and values and history and word use and vernacular that's used in that culture in order to be a true effective translator. So in other words, I need to be bilingual. And what do? We have people go study abroad. I went and studied in France for a period of time. And we do this routinely in training people in foreign languages. We don't do it in training people to be scientific translationalists. And that has to change. So we need to expose trainees to those different environments, whether it be industry, the patient foundations, the FDA, et cetera, to enrich the training experience. A number of other things we're doing, but it's very clear that this sort of single investigator working alone by themselves process, which works really well for basic science, just does not work in this space. So this is really the model here that we will have in the U.S. These hubs, we call about 60 of them, that will be supported by these centers that do central IRB, contracting, budgeting, all of this, as well as recruitment intervention innovation centers. Note that the problem with recruitment is not just finding people, it's getting people to agree to be in clinical trials. And there are groups in the U.S., underrepresented groups, who are targeting specifically to find out why they don't take part in clinical trials. And these groups have to participate as well. But the whole idea here is that the CTSA program and the RDCRN, which I'll get to in a second, would be like what we are all familiar with USB ports in our computer. That is, every time we want to do something, we don't create a new computer. We have a computer which is multifunctional, and when we want to do something, if we have a peripheral that we want to attach to it, we plug in our printer or our flash drive, or whatever it is, into this multifunctional machine, which then does magic for us. And that's really how we're envisioning clinical trials too. That the CTSA program is this giant supercomputer into which academic investigators, NIH institutes, international bodies, pharma, biotech, et cetera, can plug. Okay, so let me move on now to the rare diseases research. So you all are aware, I hope, that when NCATS was formed 4 years ago, the Office of Rare Diseases Research was moved into NCATS. And I'm going to just note here that with these little asterisks on the slides, that there are people from NCATS who are here, who I hope you will talk to if you haven't already. Steve Groff, who is the big daddy of rare diseases in the United States, is here, and even more importantly, his wife is here. And so if you talk to her and whatever you want Steve to do, that'll get done. That's probably an efficient way to do things. Am I right? Yeah. So, okay, so the Office of Rare Diseases Research. A number of things that we do, so let me just run through this. This kind of centerpiece of the program is the Rare Disease Clinical Research Network. It's a separate program from the CTSA program with a slightly different, with a operational structure. But we're bringing these two together. We actually have common leadership now under Patrick Hoffman, who many of you know, in fact. But a couple of points I want to make here is that this network is organized not around institutions, but around groups of rare diseases. And notice that in order to get supported, funded through this program, you can't work on an individual disease. You are asked to think about a level of biological organization which diseases have in common, with the thesis that these are not 6,000, 8,000 individual diseases. They're all related in some way, and we need to focus on them as all related, all related pieces in a giant jigsaw puzzle. And so you'll notice that some of the investigators said, well, they're going to study rare disorders that all affect the same biochemical pathway, for instance, like this one, the urea cycle disorder. Some said, well, I'm going to study all disorders which affect an organelle in a cell, like the mitochondria or the lysosomes. Some said, well, I'm going to study all the diseases that affect a particular cell type, like there's an inherited neuropathies, peripheral neuropathies over here. And some said, I'm going to study all the rare diseases that affect an organ, a whole organ, like the rare lung disease consortium. But whatever it is, they study between 3 and 50 diseases, and therefore are much more able to go apply insights from one disease into other diseases. So research, so progress is much more rapid than had it been organized one disease at a time. The other thing you'll notice, which is different about this, and I think responsible for that outsized productivity of this program, are these little things, PAGs here. And PAG stands for Patient Advocacy Group. A really distinguishing aspect of this program, and Steve and his colleagues this program over 10 years ago now, was that you had to have a patient group, at least one patient group involved as a member of the research team in order to get funded. I mean, this is something which is has become more common, not as common as we believe it should be, but it's become more common over the last 10 years. But this has really led the way. And I can tell you that it really has had the catalytic effect that one might have hoped in bringing focus and relevance and urgency to these programs. NCATS also supports a number of things which we're very actively looking into merging and sharing with other international resources. This is a genetic and rare disease information center that has thousands of rare diseases listed and resources, particularly in the U.S., where patients and their families can get one can actually call and talk to. It's not just electronic, and you all know that for patients and their families, this is really critical. You don't just want information, you want to actually talk to a real person. Probably true with lots of interactions we have on the phone now. We don't want to be stuck on a phone for you. We got to come on the another problem that I know all of us are interested in, and this is a hard nut to crack, but I think we have to really focus on this in the ER, is the problem of registries and natural history studies. This is a program called the Global Rare Disease Registry Program. It has a very ambitious mission, which we're just in the early stages of doing, but to have a platform whereby organizations that want to have registries can start them in a relatively facile way, and in a way which uses common data elements and make these registries comparable. Because of course, people with rare diseases and other diseases don't just have a single disease for the most part. They have multiple comorbidities. And one of the ways that we like to relate diseases to each other is by symptomatology that we're not going to recognize if we study individual diseases. So that's another program in the ORD. I'm moving back now into the early stage programs, the preclinical programs at NCATS. And we designed this part of the organization in a fundamentally different way from the way most research is done. The thesis of this organization, of this part of the organization, is that there is a tremendous amount of basic knowledge out there, for the most part generated by academic investigators. And those academic investigators are exceedingly well trained and able and smart in their fundamental science. And in my own field, I'm trained as a basic science model organism geneticist and genomics person. On the basic side, I'm a neurologist on the clinical side. But for the most part, these are folks who may have great clinical insights, may have great basic research genetic insights, but they just don't have the knowledge, experience to be able to develop an intervention. It's a whole field, this field of translation, which has traditionally lived in places like pharmaceutical companies and biotech companies, but it's very difficult for the average academic investigator to actually move a project forward on their own. And I would argue it's a fool's errand, because as we often say at NCATS, translation is an obligatorily team sport. I don't care how smart you are, I don't care how dedicated you are, I don't care how well trained you are, I don't care how hard you work, there is no single person who knows everything, from discovery of a gene, to developing an intervention, to doing clinical trials, to deploying these in the community, to be able to do this with any kind of efficiency. And so a system that rewards individual effort only, but does not reward team effort, is never going to be successful. And so the model here is that investigators somewhere in the world, and there's about 300 of these projects at any one time, get stuck, and they get stuck at various stages of the therapeutic development process. And they apply to NCATS, and depending on where they're stuck and where they need to get, they go into one or more of these programs. And then what comes out the bottom are deliverables, and these deliverables are either physical reagents, they're siRNA probes, they're therapeutics, they're small molecules, they're leads, they're repurposed drugs, whatever, what have you, and or it's data. And those programs have two essential elements to them. That is, that they need to move an individual project for an unmet medical need down the pipeline here. But importantly, they need to tell us something general about the process to make this less empirical, more predictable, so that we can do better, faster, cheaper therapeutic development. And let me just give you a couple of examples here. So let me just tell you why we're able to do this is we recruited about 80% of the people in this part of the organization come from the private sector. But you can't get hired simply by knowing the state of the art at the best places in the world. Because remember, this current state of the art was terrible. Current state of the art was to rehearse these numbers. You all know, 15 years, $2 to $10 billion, 99% failure rate. Why would we want to reproduce that state of the art? That would make no sense. But these are the folks who understand what the current state of the art is, so that you can innovate. How do you do it better? And that's really what we're trying to do. The early stage part of this is something called the NCATS Chemical Genomics Center. It does, for those of you who know about this vernacular, target the lead work. So most of our academic investigators interact with us at this level. So they have a gene or a cellular phenotype that needs to have an assay developed, high-throughput screening, chemical informatics, medchem to develop probes to test a therapeutic hypothesis or a lead for further development. And of course, we focus on unprecedented targets and rare diseases. Just give you one example. This happens to be Gaucher disease, but I could give you 100 other examples. But here it's a good example for illustration purposes. So you probably know that Gaucher disease is a lysosomal storage disease. It's a mutation of beta-glucosidase. And a number of years ago, Genzyme came out with enzyme replacement therapy, Cerezyme, which it has really been transformative for a lot of patients, but it has real limitations. One is its high cost. The second is the response is highly variable, and it's IV and lifelong. And importantly, many of these patients have brain manifestations of the disease, and this is an enzyme that doesn't get to the brain, so the brain manifestations are not effective. So an academic investigator, Ellen Sagransky, and we started working on this problem almost 10 years ago now, to develop what are called chaperones. And the idea here is that in Gaucher disease, like in many other rare diseases, there is enzyme, there is a protein produced, but it's misfolded, so-called missense mutation. And because it's misfolded, the quality control efficiency of machinery of the cell recognizes that this is not a normal protein and degrades it before it gets to the lysosome. And so there's no enzyme activity in these patients who are symptomatic. So the idea was, could we create a small molecule compound, a drug-like compound, that would bind to the glucocerebrosidase to make it fold appropriately, fool the cell into thinking that this is actually a normal protein, and get it to the lysosome, where actually, even though it's not completely perfect, it's not a normal enzyme, it has a lot of enzymatic activity, and it actually can restore function in cells. And so we first developed small molecules that can do this back in about 2007, but they were inhibitors of the enzyme in addition to chaperones. So it took us a while to develop small molecules that didn't inhibit the enzyme and acted as chaperones and corrected the phenotype in cells, patient cells. But then we ran into something which a lot of you run into, and it's become a theme that I think you'll hear about in the day, throughout the day, is that the artificial testing systems that we used in these earlier studies, particularly patient fibroblasts, don't actually reproduce the fundamental cell biology of the disorder. So Gaucher patients don't have a fibroblast phenotype. They don't have a skin phenotype. They don't have a connective phenotype. That phenotype comes mainly from macrophage problems. So we had to develop a human-based induced pluripotent stem cell model of Gaucher macrophages, which we then use to evaluate these candidate drugs. And these potential treatments have now been licensed to a biotech company in one case, and a large pharma in another for further development. So I know you just had a session on drug repositioning, so you know why we do this. But the way I fundamentally just put this is, you can pay your money and take your choice here. You can either start with 500,000 compounds and take 15 years to go from beginning to the end, and sometimes we have to do this, like I just showed you with Gaucher disease. But the other idea was, of course, that we and many others have had, is to ask, well, how many drugs are there? Could we put them all together, perhaps repurpose them in a much shorter period of time? Because one doesn't need to do all of the medicinal chemistry that I just showed you. We had to do it in Gaucher disease. So when we started this about 8 or 9 years ago, this really illustrates, I think, both the peril and the promise of translational research. I went to our informatics people, and I said, I want a complete non-redundant list of every drug ever approved for human use worldwide. So about 2006, something like that. And I thought, well, there's Google out there. They'll just go and Google it, or they'll call the FDA or something. Turns out there was no list. Nobody had ever put this together. That is, every drug that had ever been approved by the U.S., Europe, Japan, Canada, other major markets. Where were they still approved? Had they been taken off the market or not? And then looking at developmental drugs. It took the group about 5 years to do this, but now it's been done. It's a paper published a number of years ago now, 2011, and we published all the data on our website. You can download the data, and we have a collection of these drugs for, as we say, rational repurposing. And one of the things we've done with this, which has been really breakout successful, is not always rely on scientists to come to us with these problems. That we do many repurposing projects, and actually some small molecule development NCE projects from patients who walk into our office and say, patients or parents who start to work with us, and then they're the ones who actually catalyze the scientists. And here's just 3 examples. This was a program in sarcopenic tooth disease. This is a program on repurposing drugs for a rare cancer called chordoma, and this one is a rare neuromuscular disease called myotonic dystrophy. In each of these cases, it started because a patient or a parent walked into my office and said, literally, this is what the mom of the chordoma patient said. She said, I understand all the wizardry and robots and things, but I can't wait 15 years. My son has a life expectancy of 4 years. So what can we do in 4 years? And really that's what led us to doing this. Whether one is lucky enough to find a repurposed drug application or one has to make a novel treatment. And keep in mind, we actually don't know how many diseases will be treatable with one or the other yet. One thing we're really interested in. For the most part, one can't go from a lead or even a repurposed drug for many rare diseases right into into a biotech environment or a pharma environment to pick these up and take them through completion of clinical development, manufacturing, marketing, et cetera. So a number of years ago, we started a couple of programs, the biggest of which is called Therapeutics for Rare and Neglected Diseases, which does all of these things. These are things which are quite common in pharmas and biotechs, but they are very hard to do in academic environments, both because the expertise doesn't exist and the resources don't exist. And so the model here is, again, it's one of these on that big box chart I showed you. The investigators apply to us. They get put on a joint project team. They don't get money. They get put on a joint project team. The budget is given to that project team. There's milestones going into the decisions. And as long as they keep marching through these milestones, the project keeps going until it's licensed to a biotech or a pharma, or it fails. And there are two people here, I think they're both here, Liz and Sherry, Liz Ottinger and Sherry Hamilton are both here, and they work for this program, so I hope you'll, are you guys here? Ah, there they are. They're sitting in the back. You can also get to them through Jan. Jan's our molecular chaperone in this case. And there's an example here, which if you're interested in understanding how this works, this is has to be a paper that Liz, she's the first author on this paper, published a couple of years ago on a really remarkable program on human vaccine disease. It's actually freely available on PubMed Central via the National Library of Medicine. And what was remarkable about it was this, that is that this was a tremendously team effort. What you're looking at here is the group of scientists and doctors who worked in this, and we noticed that these were basic people from universities and the NIH. There were clinical people. Johnson & Johnson or Janssen made the repurposed drug cyclodextrin, so they were involved from the very beginning. The FDA was involved from the very beginning, a number of CROs, the Office of Rare Diseases at NCATS. And what Trend did is supply catalysts, which are project management and money, to make all these folks work together, allow all of them all to work together. But importantly, over here, involved in the program from the very beginning, were disease patient advocacy groups. And each of these, if you can't see the numbers, or the names, each of these little circles here is a patient group with parents and patients with NPC. And they were critical, and still are critical in the program moving forward. And so there was both a science aspect of this and a business aspect of this, if you will. So here's, like I showed you in the Gaucher example, we published a lot of this work so that others can use it. This is a study showing that both cyclodextrin, the drug that we're studying, and a version of vitamin E, delta-tocopherol, work in the cells of patients with this disorder. We then, like we did in the Gaucher example, had to move from fibroblasts to these cells that are affected in the disease, in this case, neurons. So this is an NPC model that we developed from induced pluripotent stem cells in patients with the disease. And then more recently, I've been looking at combined efficacy of these two drugs that were found up here in not only NPC, but multiple other lysosomal storage diseases. And the evidence, the data so far suggests that these two agents will be quite effective not only in NPC, but in many other lysosomal storage diseases, illustrating the point that I made before, that we've got to focus on ways to hit more birds with one stone, if you will. A big development for us happened about a year and a half ago when this program reached its milestone of being able to license the know-how and IP and the set of expert in NPC to a company. And that company, I actually didn't plan this, but this company was a spin-out of a company that went into a bag. Is Chris here? Chris Adams? Oh, there he is. Yeah, so Chris is with Cydan. This company, Vitesse, it's a misspelling of the French word for speed. It's a Vitesse without the I, so VTSSE, was the first spin-out of Cydan, which Chris can tell you is a rare disease company incubator, spinner-offer. But we have a really wonderful collaboration with folks at Vitesse now, not only us, but the folks at the Child Health Institute at NIH to move this. It's actually in an advanced clinical trial. Okay, so lastly, I'm going to tell you about one example of some of the technologies that we're focusing on that are broadly enabling. We have many of these, but this is one that you're going to be hearing about tomorrow. So I thought I'd give you a little introduction. So Dan Tagley is here from NCATS, who runs this tissue chip program. Dan's right over here. So you could talk to him, but you have to talk to him on Friday. Okay. And so make sure you don't miss that. The idea here was that many therapeutics, drugs, and vaccines fail because they do not have the anticipated efficacy that they appear to have in preclinical model cells or animals, and/or they have toxicity, adverse effects that were not seen in the animal model. So a dream for many of us for many years has been, well, gosh, what if one could develop a human-based system to test potential therapeutics? And people have tried for years to so-called humanize mice, humanize rats, take human genes and put them into rats to make them look more like humans. As we often say, lots of humans act like rats already, so that may not be so hard an effort to do that. But it actually turned out not to be as productive as most of us had hoped. But this program was enabled by recent advances in a number of technologies, in stem cell technology certainly, in biosensor technology, in tissue printing technologies, and in microfluidics. And all of those came together in this collaboration. That again, it's a collaboration like everything NCATS does. In this case, it's between NCATS, DARPA, which you may have heard about, is the Defense Advanced Research Projects Agency in the US, and the FDA. And the idea was, could you develop stable multicellular systems, organoids so-called, which represent the structural and functional elements of most human organs, and use those to study initially toxicity and potentially efficacy. So again, we'll have a human-based model, and these are all the tissues that we started with. There's some other additional ones now that have been put in place. And wow, this heart is beating very rapidly. I'm not sure why it's beating so rapidly. This is a must be excited beating. The heart is beating rapidly. But it's just an example that's easy to kind of understand. So if one is trying to model the heart, you probably know that it's actually quite straightforward to take induced pluripotent stem cells and make them differentiated cardiomyocytes. They like to do that. They like to beat independently when you do that. But it's often pointed out that heart cells which beat independently about each other is actually a bad thing. If that was happening to you would be dead. That's known as ventricular fibrillation, when the heart has contractions all over the place and doesn't work together to be an effective pump. And so how would you model that? How would you make a better model? Well, the idea here is, these are some investigators at the Beast Institute in Boston, that you can take these cells, put them into essentially a cellular brownie pan here, and then cut out a little heart brownie here, and put these brownies on a solid surface, and when they start to couple with each other, they will start to contract, and when they contract, they will start to lift off the base, and you can measure that, and that's what's being measured here. And you can see diastole systole as this goes on. And much to our surprise, quite quickly after this work was done, we see this in a number of other of the organs now. These technologies have been adapted by rare disease researchers. So in this case, some researchers down the hall from the people at Harvard who were working on the heart chip were studying something called Barth syndrome. It's a rare disease which is caused by the mutation of a drug called gene called Catalyzing. What's important here is that though this is a multi-system disease, like many or most rare diseases, these patients die of dilated cardiomyopathy, which looks like pretty much like any other congestive heart failure that patients have sporadically. And so the idea was, could you model the Barth syndrome cardiomyopathy using this tissue chip model? And in fact, that's what they did. So what you're looking at here is focus on this one down here. What you're looking at here is this is the normal heart chip, and this is what I showed you before. And you can see the contraction happening normally here. These are made from the induced pluripotent stem cells of normal people. If you take iPS cells from kids with Barth syndrome, you notice that there's very, very little contraction. So it's modeling the cardiomyopathy that you see in the disease. And over here, for pièce de résistance, it's not only can you model the dysfunction of the cardiomyopathy, but if you take the normal version of the gene that is mutated in the disease and you put it into the induced pluripotent stem cells of the kids with the disease, and you differentiate them into cardiomyocytes, you rescue the phenotype. That is, you turn them into normal heart cells. And so these are, this is a heart chip made of iPS cells from kids with Barth syndrome with the normal gene put into it. And so this gives you a very nice model for understanding why dysfunction in this gene leads to cardiomyopathy, but also a potential testing system that allows you to test. Okay, so let me just close with something that has just happened recently. And I didn't plan to have this as the headline when I made this slide earlier today, but it just happens to. It's a nice result. It's not the main point of the slide, but I'll just point out that NIH in the United States includes rare diseases in its strategic plan that just came out. This was a very important thing for all of us who are interested in rare diseases, that the overall NIH came out with a strategic plan for the next 5 years, and rare diseases really focused, really prominently featured in this plan. My main point in showing you this slide, though, is our involvement in ERGR. And just last week, I was finally cleared to be the next chair of the executive committee for ERGR. And let me just tell you why I did that, why I put my hat in the ring for them. I believe with every fiber of my being that we will only make the kind of headway that we need to make and the patients with rare diseases deserve if we work together. And rare diseases exist globally, we all know that. And so it stands to reason that we need to have a really robust effort to not just talk about how we're all interested in rare diseases, but actually have programs to actively share data and perhaps even divide and conquer the world, the whole universe of rare diseases. I should tell you, my thinking about this comes from my upbringing in the Genome Project. So many of you probably know that the Human Genome Project only got done because it was an international collaboration. The US and the local press in the UK were big members, but there were also major contributions from Germany, Japan, from Canada, from many other countries all over the world. And I experienced this firsthand. And of course, there are many other international collaborations that have happened since then, particularly in the genomics world. And we have not been as good as them in disease world are other aspects of biometric research. So the major focus of my tenure in the next 3 years is to make this happen. And I can't make it happen alone. I have you all as partners in this, but I just want to tell you how enthusiastic I am about this and how much I'm looking forward to it. It's actually the reason that I can't stay here, which I would like to have done. I'm actually going to Paris tomorrow to talk to all the folks who are involved in running and meeting with Yann and others tomorrow. So thank you. I am going to be here through about noon tomorrow, so we'd like to talk to you. Thanks.
Thank you, Professor Rustin, for asking questions, comments.
Sorry,
how do you solve the intellectual property ownership when you talk about collaborative efforts?
yeah. How do I solve the what problem?
Intellectual property.
Oh, yeah. That actually has it turned out to be less of a problem than we expected. It's always an issue when one works in this translational space, because frequently one's working somewhat almost by definition between public sector and private sector. But what we've found is that if one has a focus on innovation in this space, innovation in IP, innovation in licensing, the same way as you have a focus on innovation in science, then these things are doable. And I would just point to the partnership that we had with the TESS, which is a great example of this. It's quite a typical partnership, which preserves IP on both sides and has joint IP, but moves forward despite getting very big because of that. In the tissue chip program, it's interesting. One of the requirements of our partner in this, DARPA, and for those of you who don't recognize that, DARPA is the part of the Defense Department of the U.S. that created the internet, created the GPS, all of that stuff. So they're a very innovative group, and they have a requirement for commercialization of these platforms. And so there's already been one company, Emulate, which has been spun off from these, and there's a couple other companies that are spinning these things off. So if you wanted to make your own chips, you could hopefully eventually buy them from these companies like you buy tactile inlays from.
Hi, Alan. I'm wondering if you have like a sort of a matrix to define success. For example, do you expect from your
But the primary endpoint is a license. I think that's how people are recognized. Much like happens in biotech. The problem is multiplied though by the fact that that NHATS is not simply about being yet another organization trying to create therapeutics. There are thousands of organizations, tens of thousands of organizations worldwide trying to do that. We only take on projects when we feel that success or even failure in that project is going to teach us something generally about the problems in the translation space. So if it works, it'll be a paradigm that we can apply to other diseases. If it doesn't work, it'll tell us something about, we'll understand why it doesn't work and apply it to other diseases. So we actually have not taken on projects, we've done this quite frequently, not taken on projects because they're too good. So we've actually had to tell people who apply, this is a fantastic project, you don't need us, you should be able to get funding from the capital markets or whatever. We want a big, as one of our folks likes to say, a big, hairy, intractable problem. And those are the ones that work. Though that's what we ought to do, it does create problems with metrics, though. Because if you set metrics, and this is the danger that all of us have, if you set metrics, you gotta meet the metrics. There's never been a metric that's never been met. Why? Because people game the system to meet the metrics, right? So that's what people do. And so we actually have an innovative program in measurement, actually. We have a whole big effort on how we measure success. And we have spent a huge amount of time on that. I would say we're just in the earliest stages, but what we're doing is to approach this as a scientific problem. Say, look, it's like a scientific problem where you understand what you're trying to do, but you don't understand how you're going to get there. So we have a hypothesis of what might be good metrics, might be good success metrics, and we're going to implement those, and then we're going to watch very carefully with the assumption that those metrics are actually going to be the wrong ones, and they're actually going to have unintended consequences if they're negative. And if they do, we'll be able to measure it and change the metrics. So instead of having people change their behavior to meet some meaningless metric, we'll change the metric so that we can reward people to have the activity, to have the behavior that we want. Which is, it's actually a whole area of, frankly, of human psychology, which I knew nothing about. Turns out there's a whole field of this. They're called marketers, right? You drive down the highway, there's all these people that make all this money that, have you buy the most recent shampoo or toothpaste or whatever it is. There's a whole field of how you influence people's thinking and making do things. Translational science is very broad.
So one of the starting points was, how do we make translation of science from publications and academic laboratories more efficient?
Yeah.
And you guys pulled together all sorts of great tools, and we're a beneficiary of that. What do you think the role, however, is of getting better science being done, not just in academic labs, but also in small companies, where people run dose response curves that have positive controls and negative controls that outside groups can then compare against and run more than one animal model a certain way? True reproducibility.
Yeah. Boy, what a great question. And it is, if you listen to the question, it's actually it's a frightening question, that the question would have to be asked in the first place. Right? So you ask, how do we get people to do a dose response curve? Oh my God. That indicates most people don't know what a dose response curve is, which in fact is true. And so the implication of your question is, we believe correct, that a lot of the failure that one sees is because people are doing experiments which are not the right experiments. They're not designed the right way. They don't have controls. They're not blinded to all of these things. It comes down to this term that I heard somebody use a few years ago. It's called translatable science. There's lots of science out there, but most of it is not translatable because it's not reproducible. It's not robust. So this is something that NIH has. Well, so let me answer this two ways. One, at NCATS, our assumption is it's not reproducible. It's an artifact until proven otherwise. And we have not been disappointed, unfortunately, in that assumption so far, that most findings are artifacts, either because that's just the way biology is, stuff can happen, or because, again, it's well known in psychological circles that once a person gets a positive result, they begin to only look at data which supports their hypothesis. And this is the scientists do this the same way as other people in the culture. And there is a way to control this. So at NCATS, we have systems which do require reproducibility before we will demonstrate reproducibility before we will move forward. require in all the programs that we do that this be, or that this kind of rigor is one of the outcomes that our people at NCATS look at. And I must tell you that this is not a popular effort on the part. We get a lot of this, who are you to second guess me? I am famous Dr. Schmiegelhoff someplace, and of course, everything I do is used. And we take a very dispassionate approach to this and saying, we're not imputing anybody's abilities, but the data show us that a lot of these things might not be useful. So what it requires is something which academics, particularly, are not very accustomed to, which is something called project management. And this is one of the things that good project managers do. Incorporating project management into academic culture, again, has been a little difficult. But I'd say the reason we've been successful doing it is because as a funder, you have the opportunity and the obligation, I think, to say, if you want the money, these are the terms. If you don't want the money, don't apply. We're not forcing you to do anything. But if you're going to take the money, this is what you're going to do. But there's also a much larger effort at NIH to do this. I must tell you, though, just parenthetically, I have had conversations along the lines of the following, that it has to change within the community. I understand this, but it's pretty sense of what we're up against it. So I suggested a few years ago in a large conclave of people that if the problem is in animal studies, that the animal studies are too small, they don't have statistical significance, they have kind of three. And you ask investigators why they don't do more, they'll often say that the grant is too small. I believe the grant is 50% bigger in order to do the proper experiment. So my suggestion was we need to, at the expense of number of grants, we need to give people the resources that they need to do the proper experiment, and then hold them accountable. And the reaction was never going to happen. Why? Because politicians, at least in the United States, measure NIH by the number of grants they want. In order to do the proper experiment, we have to cut them into grants in half. And that's a non-starter. Therefore, we have to keep doing meaningless, irreproducible experiments in order to keep our funding going from time. That's a political reality. But it's something that I think as a community, Congress, I don't think, wants to do this stuff. They think this is just what they've been told that is supposed to happen. So I think it's up to us as a community to educate policymakers about the problems in this arena. Because as you said, if half of what is out there is irreducible, then there's a huge piece of it.
I'm sorry, one quick follow-up. Do you think a way to introduce this across the entire community is basically have publications say you must provide positive and negative controls, a dose response, 2 examples of the same study before we will publish, and that would force everyone to do it regardless of the cost.
Yes, and interestingly, this happened in genetics. You may know this, that in the early days of genome-wide association studies, most genome-wide association studies turned out to be false positives. And when this happened, the genetics community got together and said, and the journals, they did exactly that. You have to reproduce your finding in a completely separate group. And you know what happened? The false positive rate went essentially to 0. It's not rocket science, it's very straightforward. The problem that I think we have as a community is that the kinds of, this is a big field, and so the reviewers don't know any better than the people doing the experiments. And that's when you really run into a problem. We have not really, given the hegemony of molecular genetics in the last 30 years, we have really not trained pharmacologists and physiologists to understand these issues. And so the peer reviewers don't understand it. I think if they did understand it, they would do something about it, but they don't. Yeah, I was at a meeting not long ago, again, with a bunch of famous people this issue came up, and somebody asked, so in the group, how many of you, these were second, how many of you know how to do a power calculation? Like 3 people. And it was like out of 50 people. And these are really a distinguished group. So if you don't understand how to do a power calculation, you have major problems. You don't even understand what the issue is. So, but again, I think it's solvable with these folks.
Thank you very much, Professor Steve. So now it's my pleasure to give the floor to Yann. Maybe. So Yann Lecan is the Chief Executive Officer of Aerobies, which is the European Alliance of Patient Organizations. And we will give a talk about React for patient access to innovation. Thank you for the invitation.
It was fun to play with the word of React. Good evening, everyone. Clearly, what the kind of convention I will continue to make is from the patient perspective on these international activities and also the scientific agenda and what your priorities are in that area. So first, for the one who do not know what is the European Organization for Rare Diseases, we are an international non-profit, non-governmental group across Europe, which brings together the community of rare disease and provides a voice to influence the European and national policy in order to improve the situation of people living with this disease. So we've been founded about 20 years ago, and now we have 700 member patient organizations with pretty strict about the membership of the organization, has to be certain criteria and regular assessment. And we cover 48 countries in Europe, so we don't have members in all the European countries yet. We're covering all the EU and we don't do it. Over the years, we have created national alliances in different countries to represent, to bring together the patient groups at national level and represent them. So for the ones who are coming from the US, that's a little different with the US, where you have to structure it through each and every country in Europe and then at the European level after that. Over the years also, European federations for disease-specific group called, or specific disease or group of diseases have been formed, and that is rapidly growing, which is a good sign for science also to build a network of patient groups and patients will be better organized. We have a staff of about 40, we have over 300 volunteers participating in everything we do with the 87 languages, and our budget is shared in public and private. That's all about directors, that's the staff, just to give a sense of people, not only of the words in what we do. And you will see a series of photos in my presentation. They are all from your photo contest. They are photos taken by patients, families, or carers of patients. So putting all this into the international context, because we are very much about international collaboration in research, here I want to give you a sense of why a European organization gets involved into these international activities. And the first reason is probably because we work in these 7 languages. So the information we provide in Portuguese is available in Brazil. The one we develop in Spanish is available across all Spanish-speaking countries. The one in English obviously reaches out to Australia and New Zealand or Taiwan, etc. And also the fact that we work across countries put us already in transnational position, not international. It turns out that for these reasons, we have members in 26 countries beyond Europe that was intended. It turns out like that, so we accept them as associate members. We did fill the criteria. But more importantly, from 2008, we created the Rare Disease Day. And you know that the last day of February every year. And now there is over 80 countries, 90 in fact last week, are organizing the Rare Disease Day. And that's gaining momentum in terms of activities, and that's how we will do it. Reconnect our online patient communities, which we've created, and obviously online patient communities are not limited to the boundaries of Europe, but become immediately international. The service is available for the moment in 6 languages. We will add Russian soon, and after that, the plan is to add languages from outside Europe. Some of our activities, like the conferences, are attracting people from around the world. So there is every 2 years about more than 100, 150 participants coming from outside Europe. And the roundtable of companies where we have 45 companies, most of them are acting internationally. They are both in North America and Europe and often beyond. More recently, since 2009, we've developed partnerships, first with NORD, the National Organization for Rare Diseases, CORE in Canada, but also CORE, the same, in China, with GPA, the Japan Patient Association, which has a group on rare diseases, and the same for Russia, Russian Patient Union has a group on rare diseases, and Rare Voices Australia is specifically about rare diseases. So we develop partnership with them, which is not only to say we're together, but to have common goals. So we have these clear common goals around rare disease day, around Rare Connect, and around common activities in advocacy. So there is an alignment between our activities. And together, this patient group, we took the initiative together to create Rare Disease International. So that's something I'd like to highlight to you compared to some years ago when I went to Iraq. Is that we created this Rare Disease International as a movement to represent patient advocates and patient groups in rare diseases with a voice at the international level. And the mission of the years will be to be a strong common voice on behalf of people affected by rare diseases. The objective is to promote rare disease as a public health priority, but also as a research priority. If you read in the different slides, what is more relevant to do as already cited. And to do that, we need to do it through public awareness and policy making. So this is what we're bringing into the agenda, and I will mention in a moment how we work with the Blackburn Foundation. It's also to represent people living with rare diseases into international institutions that are not going to change their life overnight, but over the years it will become important. And we will enhance the capacity of our members. Interestingly, the International Federation on Hemophilia doesn't know what the International Federation on Epidemiology Philosophy is, the one on cystic fibrosis and thalassemia. So we don't know each other, although we basically do the same kind of work. So simply by bringing them together and exchanging information experience and developing common objectives is already arranging very much research. So last year we launched officially this Rare Disease International, that was in Madrid, in fact, also in Spain, with patient groups from different parts of the world. Some of you are in this group. And up to now, we have 31 full members, and we expect to have around 50 by the end of the year. It takes some time to go through different boards and general assembly, etc. Well, overall, we estimate that there is around 100 potential members to join this group. So only the national alliances from every country where they exist, like Singapore, Israel, China, Russia, South Africa, different countries, and international federations will do. That's the only group that can be moved. We're currently electing a council, so we have a prefiguration group of leaders, but now we're forming much more officially, let's say, this representation of leaders, and all the work is supported for the moment by the university and resources. So that's the group in Madrid last year that was together. I'm sure you will find some people you know. Now, one of the first things we did is that we have adopted a joint declaration, which is in 10 points, plus several pages of more detailed explanation, which will be progressively updated and revised. Among these objectives is the joint declaration is to promote rare disease as a public health and research priority, so clearly your goal. And the first principles are first to enhance the visibility of rare disease, to empower patients at the core of it, disseminate, create and disseminate information, improve prevention, screening, and diagnosis, access to healthcare, international networking of specialized expertise, mobility of experts and patients across countries, and international coordination of research, and shaping policy at international level. Obviously, that's the kind of agenda which is long-term agenda, but progressively we're moving on. So first, I mentioned the international institutions, and to be honest, we're not fully attracted by the UN system. I think we're all skeptic about the process and how it works. But we feel it's still an important part of what to do. It started there for AIDS, for instance. It started there for recently. So we have to start somewhere. And so we currently in this process to create a United Nations Committee for Rare Diseases, which is initiated within the ECOSOC, so the Economic and Social Council, where you have a group of NGOs, and from there it was possible to initiate that. So it's going to be a multi-year effort. We started 2 years ago. First we got the mandate to do it, then 1 year later we created it legally, and now we're in the process of formation. So the dialogue, the composition, and the first event to launch it will be within the UN building in October, November 2016. And the plan is that we will have similar events every year, alternatively in the UN building and in New York. What we want to do with that is to influence policy actions within the UN. That's the first thing that we can do, to create that visibility of the topic on the UN system and the relevant policy being developed by ECOSOC or the bodies. And that's influencing the WHO agenda, so that where disease appear on the radar, and it doesn't appear at all, it falls nowhere. And also to the fact of being into the UN system helps a lot to leverage the efforts at national level. That's very important. To help each of the international federations, each of the national alliances by saying, I'm also wearing the hat of the UN, by the way, and also the one of the WHO. So it does help. That's what it can do. On the research part, so it seems that I missed on the research part, we involve independently of Wellness International for the moment into ERGET, that was mentioned just a few moments ago. And what we're trying to do now with Wellness International, I will develop that in a moment, is to create what we call the ecosystem. So just to make sure that the different internet parties which play a role at the social level in rare diseases are doing complementary work and know what each other is doing and avoid any duplication, but rather try to align activities to enhance the potential. With the idea it's the same. For the moment in the idea we have 3 patient groups involved since the beginning. You have the US Genetic Alliance, which are theory, Nord with Peter Simon Stoll and colleagues, and Modest, several others involved. And we are in the executive committee as members along the countries, 100 companies. And also, we have patient advocates involved in different scientific committees. We have to serve as coordinator for the therapeutic scientific committee, and we have also to continue different committees. What we would like to establish in the coming months is to have a more formal relationship between Wellness International and ERGET, so to have a broader representation of patients from all over the world within ERGET, and to involve patients from other parts of the world into the scientific activities. In a similar way, we have already progressed with the Black One Foundation, thank you to Olivier in particular, and in this way at Congress, in order to establish complementary functions between what REACT wants to do, so to promote rare disease research at the international level, while we working together on the scientific priorities, but working more on the aspect of awareness and policy promotion. So that was what I was mentioning before, is creating this favorable ecosystem at the international level. Because for the moment, what existed was clearly ERGET. That's this new initiative since 2010, which we only bought since the beginning. There was also the International Conference on Rare Disease and Orphan Drugs, which exists since a certain number of years, and some others here, like Sonia, have been highly involved into it. And we want to support it, and what we have signed as an agreement is to say, we're not going to do conferences. We're going to do it only through ICOR. We're going to coordinate our efforts. We're going to bring in new people, new ideas. We're going to help you to promote it, and we organize the conferences, and then ensure that we don't duplicate efforts and need for travels and make the best use of our money. Similarly, we're very supported by the International Alliance of Patients Organizations based in London, with working closely with the WHO, so that's going to be very useful leverage. We have initiated since last year at the level of the pharmaceutical manufacturers, so that are the national associations like Pharma US or NPI in Europe and in every country around the world. They are together into an international federation that brings also all the big partners. And thanks to our work together, they have agreed now to make rare disease and orphan drugs one of their priorities, which is interesting. Internationally, orphan drugs is not really a priority, it's only to most countries. Right? So they are accepted to do that, and they're creating a working group, and we're developing a program of 3 years of what to do to create a better environment of developing the pharmaceutical and patient access around the world, focusing on some specific objectives. And we'll do the same with the other societies and with orphanet. We'll start discussion in some time to see for orphanet, international development and development of disease, we can connect it more at the local level. I've not mentioned the classification because I'm not doing the direct role, but that's a key element of these international efforts, the classification of disease. So now just to mention what do patients say about research. And I presented that in detail some time ago, I think what 2-3 years ago in that, so we'll not develop it here, but just to mention that we published in 2010 the position paper called Why Research on Rare Diseases. And that why is particularly from the work we're doing with ERARE, when it turned out, when listening to the different representatives of public funding bodies in research from different member states, that in fact, listening to them, there was nothing really convincing of why, for instance, as a research minister, it should take $1 million. So we felt, okay, we need to come up with something strongly, more strong about why to do research on rare diseases. Because if we are not able, even ourselves as patient advocates or as leaders in funding research, not able to articulate. And the following year, we developed a more comprehensive paper about what are the priorities for patients in rare disease research between now and 2020. So we're not going into details, you can go on the website and look at it. But the kind of argument of why to do research is to develop the arguments about social justice, for instance, that people with more effort that need to be made for more vulnerable citizens. So going from the high unmet medical needs, because of the life-threatening disease, the high severity, the highly debilitating diseases, and the equity and solidarity in terms of access to healthcare. Another argument was that through research on rare disease, we understand better some of the underlying mechanisms of more common conditions. The fact that rare diseases are the forefront of personalized medicine, so precision medicine, I think we could say, in fact, and for innovative healthcare policy, healthcare organization and policy. The huge success of the last years in terms of developing genetic tests for most rare diseases, the acceleration of orphan drugs approved. We see the growing number of designations, which is much more than linear, and the successful example of international collaboration. That's exactly the same before. So what kind of research? Where do we see the? So I put it on one slide, but there's 20 pages in the paper. But just to say, probably it seems that are quite obvious to you, but at least it's good because it shows a line of different patients and research. Everything around registering the biobank, or even before that, data system, where we are today, what we're asking much more, that was when we developed in 2011. Today what we would say is that we need to standardize data system of quality data through the centers of care, from which you can build registries to specific use of questions or do other research activities along the product. The link between clinical data and genetic data in the center. Allocate more funds to basic, translational, and clinical research, including from public money, which now has opened, for instance, in Europe, but that was not the case some years ago. The better understanding of some of the genetic basis in pathophysiology, but also natural history. And that's still a major issue that we have. Still now, we don't see enough natural history studies. And we see plenty of drugs failing because we don't know enough about the disease, we don't know enough about the endpoint. And the translation of research into therapies, and particularly the biomarkers and surrogate endpoints, the patient-relevant outcome measures, that has gained a lot of momentum in the last years. And we want to see much more of them, because that's really the way to speed up the development and see what the drug is doing to the patient, what is needed. All the support to study design, new study design and statistical methods. And that was mentioned by Chris too, and we see that as extremely important. So we need to really speed up some of this development. So it's not that we are against randomized clinical trials, against clinical studies, so we don't need to be capitulated on that. It's fine, but there is other options. And we need to look at these other options when studies can have much less patients involved and go faster and be cheaper. That's one of the ways to address the issue of the cost of medicine. But the problem in medicine is also that we need to see much more studies, clinical studies about the therapeutic strategies on patients and development of much more best practices of care. And social research is also very important. So to conclude, I want to stay more on the aspects of drug development because there is a lot of professional research addressed in here. We all know that the EU, as well as the US, legislative framework, which is inspired by the US legislative framework, is bringing a lot of new medicines. And we know this quickly, so I go quickly through that. But there is a big point, is that at least in Europe, we have a very fragmented landscape because of the different member states. Somehow, I observe that the US are also getting equally fragmented as a result, not only of the trend of private insurance, but also now of the implementation of the Obamacare and different rules applying from one country to one state to another in terms of access to medicine. So we face this different contexts but similar issues, I think, to a large extent. And this fragmentation in Europe is an issue in order to generate the evidence we need on the medicine. There is the fragmentation at the time of development, and the approval of the clinical trials by the different bodies in member states, that's already an issue. But you have even after approval the issue of having the common plan of collection of evidence across Europe. And the second problem we're faced with is, which is the other side of the coin of the good thing. The good thing is that drugs are being approved at an earlier stage. 50% of drugs in Europe are being approved for rare disease at the end of phase 2. And we have conditional approval, and we have now adaptive licensing. But what comes with that is that there is high uncertainty at the time of the approval. And because of these high uncertainties of the medicine, then there is a lot of access inequalities because there is a lack of trust between the payers and the companies. So what we see increasingly is that the market utilization is not anymore a yes or no decision. And what becomes increasingly important to recognize that there is a continuum between development and access. And what we're faced with is that immediately after market approval, only 1/3 of the patients in Europe get access to medicine. For the others, it takes 5 or 6 years to have access to this medicine. When you live in Romania, or in Croatia, or in Lithuania, you don't get this drug rapidly. Just to give one example, as we were preparing Rare Disease Day, that's Philip with his mother, Maria. Maria is not only the president of the Hunter Syndrome Association in the MPS in Serbia, but she's also one of the founders of the National Alliance in Serbia, which is very active. And while she was preparing the disease day, her son, Philip, passed away because of a lack of access to medicines. Treatment for Hunter syndrome was approved when he was 2 years old, but it's only 6 years later that we have access to it in this country. And that was 18 months ago. And after 12 months of treatment, the treatment was stopped for 5 weeks for administrative reasons. Nobody wanted to take the drug out of him, but because the process is cheaper. He was just within 5 weeks, in which he was at the hospital, and he didn't make it out of it. So there is consequences for this, which are very concrete, but one is it. So we have two issues that we're facing at the moment when we speak about this inflation, right, of innovation to patients, is the growing skepticism from the competent authorities, the payers, and the health technology assessors on the value of the medicines, on the value of this innovation to patients. And the second is the budget impact. Because it's one issue to have to pay 200,000 or 400,000 euros per patient per year for chronic disease, thanks to the treatment. So you have to pay the 10, 20, 30 years potentially. But it's another question also that when you multiply that by the number of patients, there is a high impact. And even though this is small overall in terms of percentage in the national budget for medicine, there is an issue of what is fair compared to the other patients and the other drugs and the other innovations. So we have a bigger issue here to solve, to bring together the actors, but also to understand what the value and determine the value, but also to find a new economic model which is affordable, sustainable. And it's not about reducing the price of the drugs by 20 or 30%, which is bargaining. It's about the model itself and development of access. And this is what I would like to conclude, is to say that we're coming here on a new deal, and that has gained a lot of momentum in the work in the last year. And what we say is that we don't want to see Europe coming into the bargaining of price of medicines simply, and only looking at the economy. No, we need to look at the science. And what we need to recognize is the continuum of evidence generation, and the fact that market position is only one point, but we need to collect much more real-world evidence from clinical use of the medicine to these patients, and then we assess regularly the value of this medicine. And that comes with another economic model, which we try to translate like that for the moment, is to say that we need at the first end during development of the medicines, and this is what I would like to draw the attention here on, is that we need, don't try to do the things too much in the classic way. Classic way can be good, but it's not always good. There's different options. Think about the other design of trials. Think about the other statistical methods. Think about doing things which make sense, exactly like Chris was saying. Is that the right question to ask? Is that the right thing to do? Is that something that is really useful and interpretable? That's the first thing. And to look much more into patient-relevant outcome measures, biomarkers, all the in vitro methods to replace animal models, all that can really help to go faster and to use much less money, invest much less in this development. Then the second aspect is to come with new evaluation process. And the key element here is the early dialogue. Early dialogue exists today in Europe with the European Medicine Agency, you are supposed to be on that, but also with health technology assessment. And now we are initiating that with payers. So companies can come very early and discuss with payers in order to align with the expectations. And then to have earlier approval. And what we are calling now, that's the next phase, is to increase European cooperation on value assessment. This is coming up, this is happening this year, thanks to collaboration of HTA. And the next phase that we now starting particularly in the economic presidency is the European cooperation on price negotiation, so collaboration between payers. And that needs to be put into the context of healthcare delivery, is that all these medicines that we're talking about here are being prescribed into specialized centers of expertise. And these specialized centers of expertise, this is where among the centers of expertise where the clinical trials are taking place. Maybe it could be called in the United Clinical Research Network. I'm not focusing on for the moment, but they might tomorrow, thanks to India. But so these centers are both performing the clinical trials, but are also, after market approval, collecting quality data on the use of this medicine for the new assessment. So progressive line between the competences of the people and the processes needed for clinical studies, clinical trials, but also after market approval and medical care. And so in this context of healthcare delivery, which is more structured and organized and higher competence, where data are key in the middle, we see a new process here of the economic model, where the discussion of the medicines will be based on value, yes, but value is a bit of an illusion, it's a curve gap. Value is one element, but there is much more on the cost. The company doesn't look only at value, they look at the money, amount of money they spend, they look at the reward to investors, they look at the strategy development. And to agree at the European level with the continuous evidence generation, what kind of research questions we have, what kind of data we need to collect real world to answer these questions, to reassess the value and the number of patients to be treated. And that comes with new type of contract and negotiations, which appears here, and that's where we are intensively active at the moment to try to connect development and access, science and economy into a new model to give to the patients within organized healthcare system for good networks. Thank you very much for your attention. These are your slides.
Thank you, Yann. Questions, comments? Sorry? You want to do? So, again, thank you very much. And I want to thank also everybody for your support in all our actions from the beginning. Again, I want to thank Ira for the wonderful collaboration on that content. It's not finished, but it's starting well, I think so. I want to thank you for your participation for this first day. I hope that you enjoy more events. And I also particularly want to thank Irdir and Siberia for their help, for the participation, to give us some very good speakers that we will enjoy the next days. And I remember that in the next meeting is tomorrow at 9 for the NGS and Alzheimer's disease. And I wish you a wonderful night in Barcelona. Sorry, Dara wants to thank you.
Thank you very much, Olivier. Of course, I was also wanting to congratulate you for the excellent conference. We are really very happy to be part of it. We think it's really an important congress and important opportunity for the researchers and for the networking. But what I wanted to say was actually about Peter and about the fact that there is initiative about continuing this networking and discussions. There is a proposition for those who would like to go to dinner for dinner together to meet in the lobby. I don't know at that time, I think so.
Yeah, just so we don't go off and have dinner all by ourselves. Maybe we can meet, maybe at registration next, you can make some dinner groups and then decide where to go. Like probably you can eat here in the hotel, but there are many nice restaurants around.
Thank you. You see that collaboration results in good opportunity. So have a good night. Je dis à Christophe,
chi non la